So far in heredity, we have learned how some traits follow a simple dominance pattern. These Mendelian traits have a dominant and a recessive trait, where the dominant masks or hides the recessive allele. Human inheritance is actually significantly more complex than this, however. There are several alternate forms of inheritance that you guys are going to learn about in this section, and it is helpful to review these as many times as possible. This video can be found on Ms. Bearcroft's Moodle page if you need to study or get some extra review on any of these topics. The first pattern of inheritance we are going to be learning about today is incomplete dominance. Last class, we learned about how a dominant trait completely covers up the recessive trait. In this case, that dominance is incomplete. It only partially covers up that recessive trait. Here's what I mean by that. When we have our heterozygous phenotype, so if you remember, that is big R, little r, two different alleles. Hetero means different. It is an intermediate phenotype. So if we had done this before with red and white flowers with Mendelian inheritance, the heterozygous phenotype would have been red. But in incomplete dominance, we have pink instead. It is an intermediate between red and white. So if you think of mixing paints, if you take red flowers and you take white flowers, you mix them together, if we're thinking about like maybe frosting or something like that, you would end up with pink. So this is a little bit different than normal inheritance. So if we look at this Punnett square, we can see a big R, that is still red. We can see little r, that is still white. But if we take a look at our heterozygous individuals, they are actually pink. And that changes our phenotypic ratio. So we used to have dominant, recessive for our phenotypic ratio. You can see it listed right here. You still do dominant to recessive, but we put our pink, our intermediate one, in between. So we had one red, which you can see here. We put that first in our ratio because that's still dominant. Two pink individuals, two heterozygous individuals, so they go in the center because they're the blend, they're the mix. And we still put the recessive phenotype last. We can see we only had one white this time, and so that one goes last. So it's a 1 to 2 to 1 ratio, 1 to 2 to 1. Let's do some practice using this new type of inheritance, incomplete dominance. I want you guys to take a look at number 1. It says, what are the genotypes of the following phenotypes? Now, during this lecture, if you want to practice, whenever I hit pause and write down your answers. And then if you hit play again, you will be able to see the correct answers. Pause. As you can see, for these different phenotypes, red has 2 dominants. White, just like we had last time with Mendelian inheritance, is 2 little r's, but the heterozygous phenotype, instead of being red, is pink. Let's take a look at problem 2. If we read the problem, it says cross 2 pink plants. What is the phenotypic ratio that you see among the offspring? Pause. So if we do these 2 pink plants, each pink plant is heterozygous, and so we can see our parents here, big R, little r, big R, little r. Let's put these parents on the side of our Punnett square. And then we can combine our 2 parents in the middle of our Punnett square. Okay, so this R and this R make this plant big R, big R. And we know that one is red. Big R, give us big R, little r over here. And big R, little r, give us another heterozygous individual right there. Those 2 are a blend and are pink. The last individual ends up being white. It has little r, and that's the end of our Punnett square. Okay, so the problem asked us to figure out the phenotypic ratio. And so we need to do the dominant first again. We had one dominant. Because the heterozygous is a little bit different, we want to record them separately. So we had two pinks, and then our recessive still goes last, so that's the 1. So again, the phenotypic ratio, we've got our 1 dominant, 2 that are heterozygous, and 1 that is recessive. Mr. Sharp is going to do the next problem on the next problem we are going to do asks us to cross a red and a white plant. We want to know what percentage of their offspring will be red.
Okay, let's look at our key. You know that the red phenotype had the genotype capital R, and the white phenotype had the genotype little r. So now, then we make our Punnett square and put in our parents, and then do our cross. We take capital R and our lowercase r, so the dominant and the recessive. We're going to cross those. You get a heterozygous. You can guess what color that is. It's a cool, different color. Then we do the same thing here with the recessive little r and the dominant capital R. And that makes another heterozygous. Same here, and the same here. And what do we get? Okay, so you see that you end up with all these beautiful pink babies. 100% of their offspring will be pink. We don't see any of the capital R, the homozygous capital R's for red, or the heterozygous lowercase r's for white. You end up with this ratio over here, and you can do 0 red, 4 are going to be pink, and then 0 are white, and we can reduce the 4 down to 1, and that tells us that all of the babies will be pink. Codominance. Both alleles are expressed in the heterozygous condition.
So with codominance, co means both or together. We have both alleles expressed in the heterozygous condition. So again, we have another form of inheritance where the heterozygous condition is different than the dominant. And in this case, both alleles are expressed.
Okay, so we have 3 phenotypes. We have the capital R, which would be all red hairs, this guy right here. And then we have capital W. We don't use the recessive R's because we have a new phenotype here, which would be all white, and that would be this guy here. And then we have a third phenotype for all you Johnny Cash fans. This is the roan color that they talk about in country songs, and that is this right here. We have the capital R and the capital white. Both of them are shown as dominant because both of those phenotypes are expressed together, and you get this roan cattle or cow right here.
So for those of you that don't listen to country songs, there are a couple other animals that show this codominance with the speckled hair patterns. So again, you have both hair colors, red and white, in roan cattle. There's also roan in horses, or agouti in goats. I used to have pygmy goats, and they had black agouti, which was black and white salt and pepper colors, and brown agouti or caramel agouti, which had both the brown and the white hair colors. You also can see this with, for example, brindle in dogs and cats.
That hat looks like a butt.
You look like a butt. Okay, we're going to do some more practice. Again, if you're doing this at home, hit pause when I say pause, try it, and then when you hit play again, you'll be able to see the answers. So if we look at number 1, it says, what are the phenotypes of the following genotypes? So go ahead and write down those phenotypes, remembering that we're talking about codominance, and both traits are expressed in the heterozygous condition. Pause. So the homozygous phenotype, red, is completely red, big R, big R. The roan is big R, big W, and it has both red hairs and white hairs, as you can see right here. 2 W's gives us a white individual. If we take a look at this problem, problem number 2, we can see that a roan bull is mated with a white cow. Okay, so again, the roan bull, big R, big W, white cow, big W, big W. If you're doing this at home, hit pause and finish your Punnett square. Okay, so the roan parents go on the side here, white parents go on the side here. Okay, so if we look at our Punnett square, we have 0 big R, red cows, 2 that are big R, W, so roan, 2 that are big W, and we can reduce that to a 0 to 1 to 1 ratio.
Okay, now we're going to cross 2 roan cattle and then ask the question, can they produce red offspring?
Pause.
Okay, so I did my cross, wrote that up here, 2 roan cattle. I put them in my Punnett square here. Okay, so we crossed our 2 codominant roans. Here we get our dominant for red and dominant for red here. That creates 1 red cow. We have our dominant for red, dominant for white here, which creates a roan cow. Same with this here, which was a roan cow. And then we end up with 1 pure white cow. So what's our ratio going to be? Okay, and then you see you get your ratio of 1 red cow, 2 that are roan, and 1 that is white. So when you explain your answer, yes, you can have a red cow show up from 2 roan parents.
Our next form of inheritance is actually one of the more common forms of inheritance. It's polygenic traits. Again, poly, we've seen this prefix many times before. Poly means many. So a polygon has many sides. If you're polygamous, you have many wives. Poly means many. So polygenic, many genes, multiple pairs of genes. Many human traits actually are the result of interactions of many different genes. Some examples include skin color, hair color, and height in humans. All of these, instead of just being tall or short, white or black, have a range of different phenotypes. So you have short and tall and all these different heights in between. If we take a look at this diagram below, on the x-axis we have the number of dominant alleles. So from no dominance to all the way up to 6. And over on the y-axis we have frequency. So how many individuals with that phenotype you see in the population. If we take a look and break down one of the genotypes, we can see there are 3 different letters, A, B, and C. These 3 different letters are each a different gene. So this hypothetical example of skin color, there's an A gene, a B gene, and a C gene, all of which you can inherit dominant alleles from your parents for. The more dominance you have, the darker and darker your skin color gets, all the way up to the extreme end over here, where we have all capital letters. That's the darkest skin phenotype. When we graph all of these, it results in something called a bell curve. So if you look at this chart, we can see that it forms this hump that looks like a bell, and the middle phenotype, the lightest or the in-between skin phenotype, is the most common because you can see, well, I can have an A and 2 capital Bs. I could have 2 capital Bs and a C. There's so many different combinations, so you are more likely to fall in the middle of this range. It's kind of like when you guys get your grades, E's, A's, and there's going to be more people in the middle. Now, that's not always the case. Skin color is a great example of this. We can shift this bell curve based on the environment. With evolution, we will talk about that. Just like in your classes, you know, you don't always have more than, you know, the majority of the people in the middle. Maybe you have a class that has a lot of idiots in it. You might have a bigger end down here. If you have a bunch of really smart kids in there, the curve might be a little bit closer to this side. But in general, it does show a bell-shaped curve.
Okay, so here we have our first problem here. Number 1, it says, what would the genotype of a human with the palest skin phenotype be? So if you look at your bell curve here, obviously down here is our palest. They have the most recessive alleles for skin pigment. Okay, so you can see that all of our alleles are recessive. We have both recessive A's, recessive B's, and recessive C's. So that'll be the palest skin pigment. Okay, in our next question, question number 2, what would be the genotype of a human with the darkest skin phenotype? Again, we look at our bell curve, and you can see the pigment as it gets darker has more dominant alleles. So this would be our most darkest skin pigment phenotype.
Okay, our last form of inheritance that we're going to learn about today is something called epistasis. So this prefix epi means above, and basically you have one gene that hides the effect of another gene. This commonly gets mixed up with just normal Mendelian dominance, but instead of alleles being dominant over each other, this is like gene dominance. And you can see we actually have 2 genes here. We have E and we have B. And this particular example is looking at Labrador retrievers. And Labrador retrievers have 4 basic colors. We have black, we have chocolate, white, and yellow labs. These 4 colors are actually controlled by only 2 genes. We're going to use the letter B and the letter E for those 2 genes. One gene, which we're going to use the letter B, determines the color of pigment. So that's either black or brown pigment. And you can see there's some black pigment in the nose of this white lab and some chocolate pigment in the nose of this yellow lab. So there is pigment present in all 4 labs. It's just these bottom 2, the white and the yellow labs, don't have it in the fur. And that's because of the second gene. The second gene, which we're going to use the letter E for, determines if pigment is produced at all. So this is similar to the gene albinism in humans, only it only affects the fur of the animal, not their skin. So we can see that this white lab has black pigment, just like the black lab, but it has this E gene, and it happens to be double recessive, homozygous recessive, which means albino fur. Brown pigment, albino fur. To recap, the 4 different fur colors present in Labrador retrievers are caused by the interaction of 2 different genes, one for brown or black pigment and one for light-colored fur. Okay, let's explore more how these genes actually work with epistasis in Labrador retriever coat color. Again, there are 2 genes. We've got gene 1 and gene 2. And in gene 1, we have the dominant allele E, which means that you have pigment produced in the fur, so this is dark all over. That is black or chocolate. We also have the recessive allele, little e, which means no pigment produced in the fur at all. That's light in color. That's like fur albinism. That's the white in the yellow labs. The second gene, we have black pigment which is dominant, it's a big B. And if we have a little b that is brown pigment, and that is a little b. You can see that the chocolate lab and the yellow lab both have little b. They both make the brown pigment. The only difference is, if we look, the yellow lab has little e, which means there's no pigment produced in the fur. So its fur is very light colored. And with brown skin under light colored fur, it makes a yellow lab. We can also see, if you get a big B for the pigment color, that means you have black pigment. And you can see that in the black lab and in the white lab. The only difference is the white lab has that little e, which again, that's the epistatic gene. It means no pigment is produced in the fur. So even though the body, the genes, every cell in this white lab's body is saying, make black pigment, because it's little e, that turns off that gene in the fur. And so it has a black nose and white fur, making it a white lab. So we can see that between these two genes, gene 1, letter E, the one that determines whether or not pigment is produced in the fur, that is epistatic. Again, epi means above. It is the trump, or the more dominant of the two genes. If you have this little e, it doesn't matter what you have for gene 2, you will have white fur no matter what. It's like a trump card if you play cards. Trump is the top value, and it covers up everything else. Okay, you will never have to do a Punnett square with epistatic genes, which is good news for some of you. But you will have to look at their phenotypes and genotypes and answer some questions. So let's do some practice. This first one says, what are the phenotypes of the following lab puppies? Pause if you're going to. Okay, so if we took it the first individual, it has little b, which gives me brown pigment. And then I have to look at my epistatic gene, and I have a capital letter, so that means I have dark fur. So it's going to have dark all over, which gives me a chocolate lab. For the second individual, we can see that for its fur pigment color, it has a big B. That means it's going to have black pigment. But it has a little e for the epistatic gene, which means light fur. Having black pigment and light fur means that you're going to have a black nose, white fur, which means it's a white lab. Okay, for the second problem, we want to know, can two yellow labs have black lab puppies? So the first thing we need to do is write down the parents for yellow labs. A yellow lab has brown skin, brown nose, so we know that it is little b, and because it has light colored fur, we know that that is for the epistatic gene, it's little e. So if you have two of those, little e, big B, you can actually answer this question without doing a Punnett square. We want to know, can they have black puppies? So let's take a look. If it's a black puppy, it has to have a big B for color. Who cares what the other allele is? It could be a big B, it could be a little b, but as long as it has a big B, it's going to be black. And then if it has dark fur like a black lab does, it has to have a big E. Okay, so that's our black lab baby. And then we also have our yellow parents. And we want to know, well, is it possible to get a black puppy from yellow parents? And if you take a look, we want to see, can this big E come from either parent? And the answer is no. Same thing with the big B. It cannot come from either parent, and so we know that this is impossible because neither parent has a dominant allele to pass on to the black lab puppies. So if you breed yellow labs, you will have yellow lab babies. That's the end of our lecture for today. If you want some additional practice, you can find more information in section 11.2 in your textbook. You can also rewatch this video as many times as you want, especially if you're having difficulties with the problems on your homework.